So let's make virology real. So this is an article from the Times yesterday. Anybody see it? FDA asks pediatricians to stop using a diarrhea vaccine. So let's see if we can understand some of this. So FDA said stop giving children a diarrhea vaccine until we can figure out why the product contains extraneous pieces of a pig virus. The vaccine is called Rotarix. It's used to prevent rotavirus infection. So what's a rotavirus? Anybody remember? Nothing?
Double-stranded RNA.
Yeah. Anything else about the genome you can remember? Is it one piece of RNA? It's in pieces, 11 pieces of double-stranded RNA. It's a reovirus. We talked a little bit about the replication of reoviruses. So this is a rotavirus, which is a big problem of causing diarrhea, especially in developing nations where they don't have IV. And the problem here is that children die of dehydration. So the vaccine will prevent that. So there are two vaccines for this virus to prevent infection. They are made by two different manufacturers, and one of them was found to have pieces of an extraneous pig virus, okay? So let's see if we can find something out about this pig virus here. All we see is it was a tough decision because we don't know if this pig virus causes disease. Here we go. See Dr. Hamburg, who is the FDA commissioner. And I think she used to be the ME here in New York City, medical examiner. She said it's a tough decision because this vaccine has been used for years and it's been safe. But this doesn't tell you what extraneous pig virus was in it. So Monday night after lecture, Dr. Silverstein and I were walking back. We were past the medical center, and we ran into one of our virology colleagues, and he said, something big is going to hit, but I can't tell you what it is. It has to do with vaccines. So I went back in my office, and I did a Google, and I found this. It was already out there. So basically, the DNA pieces they found in this vaccine, as well as in the cells used to produce it, is from this virus. Remember this guy? Circoviruses. Circoviruses have a circular DNA, single-stranded. It's negative polarity. So what they did apparently in some lab found pieces of circovirus DNA in the rotavirus vaccine. So circoviruses, well, the circovirus here is porcine circovirus. There are two types, porcine circovirus 1 and 2. Number 1 was found originally in a cell line, a pig cell line, as a contaminant years ago. Doesn't seem to cause any disease in any animal. Porcine circovirus 2 causes a disease of pigs. But we don't know if it infects people. A lot of people are actually seropositive for these viruses, but no disease is associated with them. Nevertheless, they decided to pull the vaccine because we don't know if someday these fragments might turn out to be a problem. So there's no infectious virus. There's just pieces of DNA. Okay, so far? Now, what if there were intact DNAs in this cell? It would look like this. Do you think, you should be able to answer this, knowing what you've heard, do you think this would be infectious? So in other words, the kids are given this vaccine by mouth. Vaccine goes into the intestine where the rotavirus replicates. And let's say there's some circovirus DNA in it, and it could get into cells, that's pretty unlikely in the intestine. But let's say it got into a cell. Could this DNA replicate? Is it infectious? It's a negative strand. Any ideas? So here's what you need to know. Can single-stranded DNA be transcribed to mRNA? No, it can't. Now, I can tell you another piece of information is that in the capsid of these viruses, there's nothing else except this DNA. So, do you think this would be infectious? No. Everyone says no. Anybody says yes? Well, you're not a student. yes, it would be infectious. And the logic is, of course, that if the DNA is naked in the virion, there's no protein to copy it in the virion. So this goes into cells and is repaired by the host DNA. Yes? It has to go in the nucleus, right? Without the capsid? Dr. Silverstein, if you put DNA into cells, where does it go?
Nucleus.
Yeah. A lot of it goes in the nucleus. Why does it do that, by the way?
Because that's what how it goes in. In reality, there are some instances where people introduce DNA with these lipid polymers where a good deal of it stays out in the cytoplasm. But when it's introduced as a particulate form or electroporated in, it's ingested and travels along. We don't know exactly the mechanism, but it seems to travel along filaments into the nucleus and gets taken up and is then seen in the form recognized by the cell to be transcribed and it will replicate.
So that's a good point, because in the cytoplasm, nothing could happen to this. There's no DNA repair enzyme. But a good fraction will get in the nucleus, and the polymerase will repair it. Now it's double-stranded, it can be transcribed, you can make messages, and could be. So the answer is yes, it's infectious. Okay? So there you have some real-world application of your knowledge. You could write a letter to the New York Times and say it's a good thing that this was taken off the market, because if the DNA were intact, it would be infectious. And I don't think anyone at the New York Times would know that, except you. Maybe no one in New York City would even know it. So this is just breaking. There's a lot of discussion about it. Of course, the anti-vaccine faction is now active again. So they lost Andrew Wakefield, but now they have this to complain about. So if you go, I wrote a blog post about this a few days ago, and you can see the comments already. Why can't vaccines be 100% safe? Well, the answer is that it can only be as safe as what you know to look for, right? So if you don't know every virus that's around, you can't make it 100% safe. So we'll see what happens with. I don't think this is going to be a problem, to be serious about it. I don't think the presence of DNA is an issue, but it's good to be safe. Okay. One thing from last time, remember we talked about inflammation and the 4 classical signs: heat, pain, redness, and swelling. And I just wanted to tell you that this has been known since the Roman days. There was a Roman encyclopedist by the name of Celsus who said, yeah, these are the 4 signs of inflammation. He called them calor, dolor, rubor, and tumor. So you'll hear those words. If you have a physician someday who is particularly erudite, he or she may pronounce these words, and you'll know what they mean. So those are the 4 signs of inflammation. Okay, today we're going to continue talking about the disease process. And in particular, I want to talk about how viruses cause damage in the host, how we measure it, and the genes that are involved in that. And the first thing we need to talk about is virulence. We've actually mentioned this word quite a bit, and everybody talks about virulent viruses, but not everyone really knows what it means. It's the capacity of a virus to cause disease in an infected host. If a virus is virulent, it causes disease. If it doesn't, we call it avirulent or attenuated, 2 different terms for causing reduced or no disease. We can measure this in the laboratory so that we have numbers and we can compare viruses. And there are many ways to quantitate virulence. We can measure how long it takes for an animal to die after infection. We can measure mean time to appearance of symptoms. We can measure fever. We can measure weight loss, pathological lesions. So for poliovirus, for example, you can measure the lesions in the central nervous system that the virus causes. HIV destroys CD4 positive lymphocytes, so you can count the number of those cells in the blood and use that as a measure of virulence. Many, many, many more ways to quantitate it. So here are 2 examples of measuring virulence in an animal model. On the left is infection of mice with 2 strains of poliovirus. type 1 and a type 2 strain. And here we're looking at the numbers of survivors. So we're measuring death as an endpoint. And you can see the type 1 strain doesn't do anything. The mice are resistant to it. And after inoculation with the type 2 strain, all the mice are dead by day 5. So that's just a measure of virulence. This virus is virulent, and this one is attenuated. On the right are some flaviviruses, which have been inoculated into mice. And here we are measuring a neurological lesion score. You take sections of the brain, and you look at them under a microscope, and you can see the areas where the viruses cause cell damage. And you can quantify that. You can put a number on it. So you can see in different parts of the CNS, the cerebrum, the brainstem, the spinal cord, the different flaviviruses cause different extensive lesions. So some cause a lot of lesions in all places. Other viruses prefer the spinal cord, and dengue doesn't cause many lesions anywhere. So just another way of measuring virulence. Now, virulence is relative. It's influenced by many parameters, including how much virus you put in, where you put it in, the kind of animal, and all of that. So you cannot compare virulence if you're using different systems. You can't compare smallpox and poliovirus because they're measured in different ways. The virulence is measured in different ways. Okay, so you have to always remember that. It's totally irrelevant to compare two viruses in terms of virulence unless they're assayed in exactly the same way, and they never are if they're totally different viruses. And if you have two polioviruses, the only way you can say polio 1 is more virulent than polio 2 is to assay them exactly the same way. Okay? Now, here's an example of how the route of inoculation in the animal will affect virulence. These are mice inoculated either subcutaneously beneath the skin or intracerebrally right into the brain. They're both suckling mice, that is, they were just born. And on the x-axis, we have days after infection. And on the y-axis, we have here on the left, we're measuring virus in the serum and in the brain, or in the brain or in the serum. So this is PFU per milliliter or microliter. And here we have PFU per milligram of brain. So we're measuring the virus in the brain here. So you can see on the left here, if we inoculate La Crosse virus subcutaneously, we measure virus in the brain. It replicates quite nicely. The viremia is a little lower, but you can see you get a nice viremia. But a different strain of the same type of virus, Tanya virus, doesn't cause any virus replication in the brain or any viremia when inoculated subcutaneously. But if you put the virus directly into the brain, you see that both La Crosse and Tanya both replicate well in the brain. Okay, so where you put the virus matters in terms of virulence. And here's another example of the effect of the route of inoculation. Here we're looking at the number of virions that you need to kill half of the infected animals. So these are suckling mice or adult mice. And two different viruses. We have a wild-type La Crosse, which is a Bunya virus, by the way. When inoculated either intracerebrally or sub-Q in suckling mice or IC in adult mice, you just need one PFU to kill half of the mice. It's quite a virulent virus, right? But in sub-Q in adult mice, it takes 10 times as much. Now, in comparison, we have a virus mutant, which by IC is quite virulent, 1 PFU to kill half of the animals, but you need over 100,000 by the sub-Q route, over 1 million, or over 10 million in adult mice. So not only the route, but the age of the mouse and the type of virus will affect the virulence. So these are just some very simple examples, but you can imagine that this can get very complicated if you don't control for every parameter. So you have to compare viruses in the same way. So in virology, one of the major efforts is to identify what controls virulence. Because if we can identify what causes it, maybe we can interfere with it. So we want to know the virus genes that are involved, and we want to know what host genes are involved with it. And we do this by genetics, typically. We make mutations. In the virus, we can make mutations now in just about every viral genome. and we can ask the effect on virulence. And we can now do the same in cells. We can target genes specifically in cells. So here's an example of making virus mutants and some of the problems that arise when we're trying to interpret the results. So here we have a wild-type virus. It grows very well in cell culture, and that's shown by making nice-sized plaques. If you inoculate this virus into the brain of mice, it kills them, so we say it's neurovirulent. So you want to know what genes of the virus are involved in killing mice. So you make mutations in the virus, and here are two possible outcomes. Here's one mutant virus that grows very poorly in cell culture, only a few small plaques. And you put it into the mouse brain, and it's attenuated. It's not virulent. But this virus is uninteresting because it's just sick. It doesn't grow well, and that's why it doesn't cause disease. It doesn't tell you anything about a specific gene involved in virulence. All it tells you is that to be virulent, you have to grow well or replicate well, but that's not particularly interesting. The virus here is more interesting. We've made a mutation in a gene of the virus. The virus grows well in cell culture, but when you put it into mice, it doesn't cause disease. It's attenuated. That's interesting because it's specifically required for virulence, not just for efficient replication. So these are the kinds of mutants we like because they tell us about genes that you need to cause disease. So what kinds of genes are these? This has been done in many laboratories on many different kinds of viruses, and you could put all the genes identified so far into 4 different classes, the genes that affect virulence. First, we have the ones that affect the ability of the virus to replicate. And as I said, these are not terribly interesting, so we don't pay much attention to those. More interesting are genes that modify host immune defense mechanisms, and Dr. Silverstein will tell you more about those. These you can take away from the virus, and it grows perfectly well in a cell culture because cells in culture don't have immune systems. And in an animal, they will be less virulent. There are also genes that allow the virus to spread within a host. You remember, viruses have to go from one place to another to reach their targets. So those are interesting genes. And then we have a few genes that have intrinsic cell-killing effects. That is, they damage cells, and that's why they cause virulence. So let's look at a couple of examples of such genes or such sequences that have been identified. And the first are actually not in genes. They're in non-coding regions. So these are non-coding sequences that have been shown to affect virulence. The vaccine strains of poliovirus, they're called the Sabin vaccine strains. These are infectious strains that you take orally and replicate in your intestine. They don't cause polio most of the time, although some of the time they do. They have a mutation in the 5' non-coding region that reduces their neurovirulence. And in another virus, mangovirus, which is a mouse virus, deletions within a poly-C tract in the 5' encoding region will also reduce virulence. These mutations don't affect growth in cell culture, only in an animal. So here is the mutation in poliovirus that attenuates virulence. I don't know if you remember, the 5' end of poliovirus RNA is a non-coding region. It's quite long, and it contains this highly structured RNA, which is called an iris, an internal ribosome entry site. And if you remember, that allows ribosomes to bind internally. This is composed of many stem loop structures, as you can see here. The vaccine strains of polio, the Sabin vaccine strains, have a mutation in this specific loop right here, which is blown up on the right here. There are 3 serotypes of poliovirus that are in the vaccine, so it's trivalent. And there's 1 base change in this region in each serotype. This is the type 1. It goes from there's a G here, which goes to A at this position. This is the change in type 2, G to A. And then there's a change in type 3 at this C to a U. So slightly different changes in all 3 vaccine strains, but the effect of all of them is that they make this a vaccine. It makes it safe for us. So what do these changes do? Here's an example of what they do in a mouse model. So you inject mice intracerebrally with poliovirus. It replicates in the brain, and it causes paralysis. So let's look at a virus with a C at 472. So 472, go back here, is the position in the type 3 strain of polio, which is changed in the vaccine. The vaccine has a U. So if you put a virus with a C at that position, you can see it's virulent in mice. The amount of virus you need to kill 50% of the animals is about 9,000 PFU. If you change that to a U, and this is the only change you make in this virus, the LD50 is now, there's no endpoint. You can't kill any mice by putting as much as 10 to the 7th PFU directly in the brain. So one base change has done that. And if you look at the replication of these viruses in mice, so we're looking now at days after infection and the amount of virus per gram of brain, you can see viruses with a C multiply quite well. If you change that to a U, they don't replicate at all, they're cleared. So viruses with a U don't cause paralytic disease, and they don't replicate well in the brain. And this is why when you get this polio vaccine, it doesn't paralyze you, because of this one base change in the 5' non-coding region. How about some genes that modify host defense mechanisms? Well, Dr. Silverstein will regale you with these next week. Let's just give you an overview. Many viruses encode proteins that mimic cytokines. You remember the cytokines are important in the immune defense. They cause inflammation. They recruit cells to the infected area to try and clear the infection. And many viruses make homologs of them that will bind the normal receptor for the cytokine on whatever the host cell is and not signal. They also encode viroceptors. They're homologs of the cell receptors for cytokines. So they soak up all the free cytokine that's produced. So they impair the host immune response. They mimic normal cellular molecules that are critical for host defenses. And they do it at both the innate and adaptive levels. The interesting thing here is that these are not required for growth in cell culture. You can take these genes away from the viruses and the viruses will grow perfectly well, but in animals they're quite attenuated. Most of them have been found in the large DNA viruses, as you'll hear, and that is because these viruses have a lot of room in their genomes to encode these proteins. But these are viruses that kill cells very efficiently, and they induce inflammation. So they have been evolutionarily selected to produce these antagonists. Otherwise, the immune defense would clear all the infections. So some of the examples which you'll hear about: soluble cytokine receptors, proteins that bind parts of the complement cascade that we discussed last time, and also proteins that affect antigen presentation, the presentation of viral antigens to cytotoxic T lymphocytes or B cells. Viruses block that. So you can't make an immune response against them. Very clever. So here's an example of one such gene. It's in a herpes virus. It's called M3. And what we have here on the left is a survival curve for wild-type virus in mice. So we have increasing amounts of virus inoculated into the mice. And here is the dose response. You can see with high amounts of virus, most of the mice are dead. And if you take this gene away from the virus, you delete this gene, which encodes a chemokine receptor. Now the virus is not as virulent. The mice survive with higher doses. Eventually, they will all die with 1,000 PFU, but you can see the curve is quite different. So this simply means that there are other such genes in the genome they haven't gotten rid of. Now, this is a chemokine receptor. What do chemokines do? They signal cells to come into a region and infiltrate and help clear the infection. And you can see that this deleted virus, the virus lacking the chemokine receptor, is unable to prevent the infiltration of macrophages in this animal, for example. So these are different cell types that are measured infiltrating into the infected area. And the blue is the wild-type virus. If you take away this chemokine receptor, you see the virus is not as good as inhibiting infiltration of macrophages. And that's exactly what you would expect from an antagonist of a chemokine. And there are also effects on other infiltrations as well. Genes that enable virus spread. Two examples: reoviruses. We just talked about a reovirus, the rotavirus, earlier. They encode a protein called capsid sigma 1, which is important for determining how the virus spreads. If you put virus intramuscularly into mice, it spreads via the blood to the CNS. If you put a different type of reovirus in, it spreads by the nerve. And the gene that controls this route of spread is the capsid protein sigma-1. A single amino acid change in a herpes virus, glycoprotein, also influences the spread of the virus in the central nervous system after you put it in the foot. So 2 examples of viral proteins that affect spread. Some viruses encode proteins that have intrinsic toxicity. This is not, yes?
I have a question about how they determine how they should put in,
How you should inoculate a virus? It's usually empirically done. It was done years ago. You find an animal that's susceptible to your virus, and you determine which route is useful for your purposes. So you may have a particular virus. Let's say you find that a mouse is susceptible to your virus. You put the virus subcutaneously. There's no infection. Then you look elsewhere. You try intramuscularly, you try intracerebrally, and eventually you find a route that works. You'd like to mimic the way the virus is brought in, but you can't always. So for flu, you would like to have the virus delivered to the respiratory tract. For polio, you would like it to be ingested, but that's not always possible, so you have to inject the viruses. So it's an empirical approach. You find what works, basically.
Are there specific properties about a virus that make the way that you inoculate something? Like, are there specific characteristics of a virus that would make one method of inoculation better than the other, or it just always has to be done?
Well, for respiratory viruses, you prefer to put them in an aerosol. So influenza, measles, respiratory syncytial virus. There are animal models for those, and you can spray the virus or drip it in the nose. But for other viruses, like herpes viruses, correct me if I'm wrong, which you would normally acquire at birth, they have to be injected in mice. So it's not approximating the natural route. So you don't always mimic the natural route of infection. You can't. Yes?
Does changing the gene within a virus that causes it to go to another site other than its usual site of infection causes different pathogenic effects on the site that it infects?
Often, yes. So you can make a virus do things that are very different from what it does in a natural host by changing its tropism, sure. Yeah. Okay, so this is an example of a virus protein that's toxic. We don't usually have such proteins in viruses. Bacteria, on the other hand, make toxins that make you sick, but viruses generally don't do that. So I want to give you one good example of this. This is a protein in rotavirus. So rotaviruses cause diarrhea, which means you have a fluid imbalance in your intestines. And that's caused by this protein called NSP4, which is produced by the virus. If you just make this protein in cells, it causes an increase in intracellular calcium levels. So that gives us a clue about how this protein is working. If you feed the protein to mice, the mice get diarrhea. So no virus, just give them this protein. So basically what happens is the protein makes cells secrete sodium chloride, and then you cause a fluid imbalance in the intestine. You stop absorbing the water that would normally be absorbed because there's too much salt in the lumen, and you get diarrhea. So that's all done by NSP4. And the protein, so it interacts with transport molecules in the plasma membrane. This is the epithelium of the gut. It causes a rise in calcium levels, and it also triggers a signal transduction pathway that results in excretion of sodium. And so that's what causes the diarrhea, this one protein. How about this cell? There are certainly genes in the cell that contribute to virulence. And now we can disrupt these and ask which ones are they and what's their effect on virus infection. So they're virulence determinants. And all of the host genes that are needed for translation, genome replication, mRNA synthesis, these are technically virulence determinants because you can imagine disrupting the genes and the virus would lose virulence. The problem is they're also required by the cell, so it's very difficult to study these kinds of genes. There are also microRNAs, very small RNAs in the cell that have regulatory processes. These have just been discovered very recently, and these you can disrupt or remove and show that they have an effect on virus virulence. So here's an example of one cellular protein that's a virulence factor, TRIM5 alpha. Old world monkeys don't get infected with HIV-1. And the reason is the virus gets into their cells, but there's a block before reverse transcription happens. So you never get a DNA copy of the genome and it never integrates. The reason there is this block is because of this protein made by old world monkeys, TRIM5 alpha. This protein binds to the viral capsid and somehow prevents reverse transcription. We're not sure how it does that. And if you remove TRIM5 alpha from old world monkey cells, you can do this in a variety of ways, you relieve the block to replication. So HIV will grow in old world monkey cells if you take away TRIM5 alpha. So it's a virulence determinant. MicroRNA is very interesting. Here's a very neat example. There are many, many different microRNAs in all different tissues. And this one is called miR-122, microRNA 122. It's a liver-specific microRNA. It's needed for replication of hepatitis C virus. So if you take this away from cells, hep C will not grow very well. So this is a virulence determinant. We don't know exactly why the virus needs this for replication. The mechanism hasn't been sorted out. A paper was just published a few months ago showing that you can remove miR-122 by giving cells or animals a complementary RNA, which basically makes the miR-RNA non-functional. And in one study, this was given to chimps, which were then infected with HCV, and they had significantly lower viral loads. So this is a very interesting new approach to antivirals. HCV has been very difficult to treat, and this may be a good way to do that. So now let's move into talking about cell injury and how viruses cause it. We've talked before about the so-called cytopathic viruses, the ones that kill cells in culture. They inhibit macromolecular processes of the cell. Membranes break up, enzymes leak from lysosomes, the cytoplasm degrades, you get syncytium development by envelope viruses, you get induction of programmed cell death. These are all ways that these cytopathic viruses kill cells. And this is all a major component of the virulence of a virus. That is, what a virus does inside you for many viruses is related to all of these properties. But many viruses don't do any of those things. They don't cause any cell damage. And it turns out that the immune response is what is responsible for the pathology of infection in these cases. We call that immunopathology. And as you know, of course, many of the clinical symptoms, the fever, the rubor, dolor, calor tumor, is an immune response to virus. So the aches and pains, the fever, the nausea, that's not the virus doing something specific. It's you responding to the infection. And for many viruses that don't kill cells, the immune response is what's causing the pathology. So let's have a look at that. Here are a number of different viruses where the disease is caused by specific components of the immune response. So we have CD8 T-cell mediated disease for these viruses. We have CD4 mediated disease, and even divided into Th1 and Th2 subsets of CD4 cells. And there are even diseases caused by antibodies, and we'll see by cytokines as well. So let's take a look at some of these examples. Here's an example of a disease mediated by CD8 positive cytotoxic T lymphocytes. Remember, those are the lymphocytes that kill infected cells. So this is a mouse model for lymphocytic choriomeningitis virus. This is a virus of rodents. It's of really no consequence to people. But if you do have a pet hamster, it's likely to be infected with LCMV. And you'll probably be all right unless you get immunosuppressed for some reason. An organ transplant, if you get AIDS, then this virus can kill you. It's been isolated from people who have died after organ transplants with disseminated infection. But it's a great lab model for studying immunopathology. So if you inject mice with LCMV, they die within 8 days. But if you infect mice and immune suppress them with some steroid or other immune suppression, the mice live and they become persistently infected. That is, they produce virus, but they don't have any immunopathology. So you have suppressed some aspect of the immune response that's responsible for killing them. If you now give these mice CTLs from another mouse that has recovered from an LCMV infection, so those are virus-specific CTLs, the animals die. So that shows you that the CD8-positive CTLs are causing the death. They are causing the immunopathology. Other examples of immunopathology associated with CD8-positive T cells, myocarditis caused by Coxsackie virus. An infection of the heart often leads to you needing a heart transplant. It destroys the heart muscle. This virus in heart tissue doesn't kill the cells. It's non-cytocidal. It only causes damage in people with good immune systems. And the idea originally arose that CD8 cells were lysing the myocytes, the cardiomyocytes, killing the heart cells, causing the damage and trying to clear the infection. So what was done was to make mice lacking the gene for perforin. Remember, CTLs have two important ways of killing the host cell. They have perforin to poke holes in it and then granzyme to digest the cell. So if you make mice that lack perforin and infect them with this virus, they develop less severe disease. So it shows that CTLs are important in development of disease. And they also may release cytokines that recruit other inflammatory cells to the area, and that may contribute to the disease as well. For example, if you take a chemokine gene from mice, you also reduce disease after infection. So the pathology is not only due to CTLs, but also to chemokines. So here's a slide which shows that. So these are two sections of heart muscle from mice. On the left is a wild-type mouse. It's infected with Coxsackie, and then it's stained with a particular dye which shows up blue here, and that shows the dead cardiomyocytes, basically. And on the right is a mouse lacking the perforin gene. It's been infected with the same virus, Coxsackie, and you can see there's no damage after staining. So just taking away the perforin gene really reduces the immunopathology. Liver damage caused by hepatitis B virus. If you make transgenic mice that express the glycoprotein of hepatitis B, they're fine. The mice have no problems whatsoever. But if you put virus-specific CTLs into those transgenic animals, they develop hepatitis. So it shows that the CTLs are responsible for hepatitis. They attach the hepatocytes, they induce apoptosis, and they kill them. Also, of course, there's cytokine release, which contributes to the liver damage. If you put, remember we showed this experiment last time, if you put anti-interferon gamma into these animals, or if you take away the macrophages, you prevent the immunopathology. So those are two components that are contributing to that. CD4 cells can also cause immunopathology. Remember, these are the helpers of two different subsites. They make a lot of cytokines. They can recruit cells into infected areas. So you can get what are called delayed type hypersensitivity responses. That is when cells, T cells, come into an infected area and overreact, produce too many cytokines, and you get very intense inflammation. The kinds of cells that are recruited by CD4s are mostly neutrophils and monocytes, which cause tissue damage. And they release a variety of compounds to do that, cytokines, TNF-alpha radicals like nitric oxide, and proteases. So the CD4 cells secrete cytokines that recruit neutrophils and monocytes, which release the things that damage the tissue. And we have examples of both Th1 and Th2 specific immunopathology. So Tyler's virus is a virus of mouse that causes demyelination. It destroys the myelin sheathing around your nerves. And we think that Th1 cells are involved in this. We think that they recruit phagocytes that in turn degrade the myelin sheathing of the nerves. And we believe that those macrophages release superoxides like nitric oxide and other radicals that do the damage, as well as secreting cytokines that destroy the myelin sheath. In many diseases, you get rashes or poxes, and these are often delayed type hypersensitivity reaction. In other words, an overreaction to an immune stimulus. Now, none of you have probably had a tuberculin test in your life, but if you want to know if someone has been infected with tuberculosis, you inject a little bit of tuberculin antigen into your arm right here, and within a very short time, you get a delayed type hypersensitivity reaction if you are infected or were previously infected. And what you're getting is an infiltration of cells into the region caused by cytokines released from CD4 cells. And many virus infections make a rash, which essentially is a delayed type hypersensitivity reaction. Measles, smallpox, varicella zoster. And in these cases, the Th1 cells and macrophages home in on the infected foci and secrete cytokines and cause this delayed type hypersensitivity reaction. So these are some of the cytokines: IL-2, which we just heard about today in the seminar, and interferon gamma. And again, these cytokines cause your hypersensitivity. They make a little induration or bump. They mess with capillary permeability, and that's how they cause that reaction. Herpes virus infection of the eye, a very common cause of blindness, almost entirely an immunopathological reaction involving CD4 positive Th1 cells. So what happens here, you get infected. Many times you eventually will have blindness. And the virus replicates in the corneal epithelium, and CD4 positive Th1 cells come into the infected area, and they start to recognize the underlying stromal cells. So they don't recognize actually the corneal epithelium itself, the outer layer. They recognize the layer below it. They're recruited there by the infected cells. And in fact, these CD4 cells come into the area after replication of the virus is done. So they're attracted there by the initial cytokines released by virus-infected corneal epithelial cells. Those cells then release cytokines that attack the stromal cells and eventually leads to blindness. So it's again an example of an immunopathological reaction. Th2 cells, remember, help mature B cells to mature. Respiratory syncytial virus is another non-cytolytic virus. So again, the pathology in people caused by the immune response. A very important lower tract pathogen in infants. This is a virus that preferentially infects babies within the first 6 months of life. Very difficult virus to control. If you immunosuppress mice and infect them with this virus, they get less severe lung damage. And if you give those mice antigen-specific Th2 CD4 cells, you make more lung damage. So again, there's another way of showing that a specific subset is involved in immunopathology. The lesions that you see in mice have a lot of eosinophils in them. So we think that some of the cytokines produced by the Th2 cells are recruiting eosinophils to the lung, and that causes the RS virus pathology. In many infections, there is normally a very precise balance of Th1 and Th2 cells. We alluded to this a little bit last time, how the particular cytokines produced by the infected cells control this. And this is very important for respiratory syncytial virus. Now, in a kid infected with RS virus, the T cell, the CD4 response is mainly Th1, right? And this is a protective response. Many years ago, a vaccine was produced against RS virus, and this is a very important virus to prevent, so many efforts have been made to make vaccines. This vaccine was made by inactivating virus with formalin. So it was an activated virus preparation. When this was used to immunize kids, they got a Th2 response, not a Th1 response. So they had an aberrant CD4 cellular response. Those kids were still susceptible to infection with the virus. So there were clinical trials done where children were immunized. They go back out into the world, and then they would get infected and get even more severe disease than they would have gotten if they were not immunized with this formalin inactivated preparation. And these kids with more severe disease have many eosinophils in the lung. So those are the cells which we think are causing the pathology. We now know that by inactivating this virus with formalin, we destroyed its ability to activate the innate system. Specifically, the wild-type virus is recognized by TLR2 on the cell surface, and that leads to cytokine production, which will make a very high affinity antibody response eventually. If you inactivate the virus with formalin, it no longer activates TLR2. You don't get this cytokine cocktail produced, and you get the wrong balance of Th1 versus Th2 cells, and the kids develop more severe disease. Okay, so this is a great practical example of how you can mess up the balance of Th responses by breaking a virus with formalin. Cytokines can also cause immunopathology. You may know this as systemic inflammatory response syndrome or cytokine storm. When an infection goes over the threshold initially, remember the innate system can't handle it, you have an amplification of the immune response and engagement of the adaptive response, you make a lot of cytokines, and you have a global inflammatory response against infection. And if this happens too quickly, say out of proportion to the infection, you can release more cytokines than needed, and you have a systemic inflammatory response, which can be pathological. This often happens when a new virus crosses from animals into people. So Ebola and Marburg viruses typically cause these massive cytokine eruptions in the host. So these are viruses that haven't co-evolved with us for many years, so we haven't learned how to balance their infection or our response to it, cytokine storms. And we believe this might have been partly responsible for the lethality of 1918 influenza, but we don't know. And other microbes do the same thing, toxic shock syndrome or toxic sepsis caused by a variety of bacteria. Same mechanism, overproduction and dysregulation of cytokines. Here's an example of a cytokine that's involved in a viral pathology. This is West Nile encephalitis. West Nile is recognized by toll-like receptor 3, which senses the viral RNA. And we have a mouse model for West Nile virus where you can inject the virus and it goes to the brain and causes encephalitis. And what was found is that if you take the gene for TLR3 away from mice, you make TLR3 knockout mice, they are more resistant to infection than wild-type mice. So they are less likely to develop encephalitis. And they don't make as many cytokines as wild-type mice, which makes sense because TLR3 senses the infection and signals cytokine production. It turns out that one of the cytokines that would be produced in a wild-type mice is TNF-alpha. And this is a cytokine which, among other things, compromises the blood-brain barrier. So, you know, there is a barrier between the circulatory system and the brain tissue. It's a permeability barrier. And this cytokine loosens the junctions so that things can pass through more easily, including viruses. So the West Nile infects, TLR3 senses it. In the brain, the virus now can get into the brain more easily and cause encephalitis. So if you take away TLR3, the mice actually are more resistant to disease. And this is a very nice experiment which shows this. These are the brains of mice that have been infected with West Nile virus. And this is 0, 6, 12, and 24 hours after infection. And what you do in this experiment, you give them virus, and then just before you sacrifice them, you inject them with a dye, which normally doesn't get into the brain unless the blood-brain barrier is compromised. So you can see by 24 hours, the dye is in the brain of the West Nile infected mice. Now, the real experiment, the TLR3 knockout experiment, isn't shown here, but in that experiment, there's no dye infiltration into the brain after infection. So just taking away TLR3 prevents that from happening. Now, this is not to say that we should take away our TLR3 to make us more resistant to West Nile encephalitis, but we have to figure out ways to intervene knowing that this could be a problem. Here's an example of antibody-mediated immunopathology. So we went through the T cells, cytokines, and now antibody. Dengue fever caused by dengue virus, a mosquito-transmitted flavivirus, Aedes aegypti. And this is endemic in many, many parts of the world. Billions of people are at risk for infection with this virus. There are about 50 million infections a year, a really important cause of morbidity. And here's a map showing you the distribution of the vector, Aedes aegypti in yellow, and in red the areas infested with dengue virus. And this has been spreading over the years. So you can see from here to here prior to 1981, and afterwards the virus is spreading southwards. And part of the reason is that the vector is being spread in the used tire trade. There are many big container ships that ship used tires all over the world, and as I said before, they always have water in them and they carry the mosquito larvae with them. You'll hear more about this from Dick Despommier at the end of the course. So the virus causes dengue fever. In most people, this is not a problem infection, and you can be asymptomatic, or you can get the standard symptoms of virus infection: fever, pain, redness. What was the other one? And you can recover and you're fine in 7 to 10 days. In a rare number of infections, you can get a hemorrhagic fever where the virus breaks your capillaries, and this can be a life-threatening disease, and you can bleed to death. This is what's known as dengue shock syndrome. You make antibodies after this first episode, but the problem here is that there are 4 serotypes of virus, and the antibodies against type 1 don't protect against type 2 and type 3 and type 4. But if you get infected with another serotype, the antibodies will bind to that virus. They won't block its infectivity. They will not neutralize its infectivity. But they will now allow the virus to be taken up into cells with Fc receptors on their surfaces. The Fc receptor binds the antibody, and the virus is attached to the antibody, so it now can get into, say, macrophages, which it normally would not get into, and destroy them. And the macrophages release cytokines, and that causes dengue shock syndrome. So you now have the replication of the virus in the wrong place because of the antibody allowing the virus to get into those cells. So these are non-protective antibodies that enhance infection by Fc receptor-mediated uptake, cytokine release, and severe symptoms. And the incidence of the shock syndrome now goes to 1 in 90 or 1 in 50 as opposed to 1 in 14,000. So you can see why this is a big problem. There's a lot of dengue in those areas to begin with, so there's going to be a lot of reinfection. So people are working very hard to try and prevent this. There are other ways you can get immunopathology with antibodies. Immune complexes can form. So if you get infected with a virus and you make antibodies, these two combine. And if the process is not regulated properly, if too many viruses and too many antibodies combining, they can make deposits in very sensitive parts of your body. So in the kidney and in the brain, they're very, very fine capillaries, and these complexes will deposit in the capillaries and cause problems. So those are caused immune complexes, where you get a lot of virus and a lot of antibody response, and the complexes are not cleared. And this causes a kidney infection, glomerulonephritis, inflammation, vasculitis, and I like this one, mental confusion, if the complexes go up here. But I don't think we all have immune complexes. There are other reasons for being mentally confused, including me. Free radicals can cause tissue damage. Remember nitric oxide we talked about last time is produced by an interferon-inducible enzyme, nitric oxide synthase. It converts arginine to nitric oxide, and then that gets attached to proteins and inactivates them. So the idea is that this gets attached to virus proteins and inactivates the virus. So the way this is made is that if you have an epithelial layer infected with viruses, remember they're secreting cytokines and chemokines. Immune cells like macrophages are recruited in the area, and they produce nitric oxide. The idea is to kill these cells so that the virus replication is stopped, but in the process you get immunopathology. And it's been shown in a couple of virus infection models of mice that if you take away the ability of the mice to make nitric oxide, you actually get less immunopathology. Now, there are a couple of pathologies associated with virus infection that are slightly different. I want to go over them. One of them is autoimmunity. Many infections cause autoimmune disease. That is, you start making antibodies against yourself. And this has been amply showed in laboratory animals, but we're still not sure if there are bona fide virus-triggered human autoimmune diseases. There's a lot of evidence. I'll show you some of it now, but it's still quite an open question. There are a couple of ways this can happen. We have in our bodies anatomically sequestered sites that are not normally exposed, and maybe viruses can damage them so now that the antigens are no longer recognized as self, and you get an antibody response against yourself. Or you may unmask antigens that are normally not seen by the immune system, by antibody complexes, by release of proteases, and so forth. Or there is molecular mimicry. That is, an epitope in the virus is the same as some cellular protein that you have. So you share antigenic determinants, and then when you get infected, you make antibodies against the virus, and some of those cross-react with your host tissues. And many peptide sequences are shared. If you just take all the known sequences of viruses and the human genome, you can find quite a few that cross-react. An epitope isn't very long, so the chances are quite good of getting cross-reactivity. This has been hard to prove, but we can really show this quite well in transgenic mice. So for example, if you make transgenic mice that produce individual proteins from LCMV, these mice are fine. So these are, the mice make the proteins from birth, so they're recognized as self. They're not known to be virus proteins any longer. But if you infect those transgenic mice with the virus, they develop disease, which is an autoimmune disease. Now you're making antibodies or cellular immunity against the virus determinants, and it's pathogenic. So somehow the recognition of those antigens is regulated in the absence of virus infection, and then virus infection dysregulates that to cause the autoimmunity. And the virus-specific CTLs are at fault. So one idea for how people develop autoimmunity is that you're infected at birth with a virus, it persists, but you don't recognize it, and then if you get infected with the same virus later on, you get autoimmunity, similar to the mouse model. We don't know why, if you're infected at birth, you remain tolerant. It has to do with the number of T cells, how high their affinity is for the antigen, and so forth. There are a lot of issues involved that we don't quite understand. Here are some of the epitopes that are shared among virus and host proteins. So here, a poliovirus protein. You see, this is an epitope, quite short, and there are some conserved sequences with the acetylcholine receptor. Papillomavirus and the insulin receptor, rabies, HIV, measles. So in animals, you can show that most of these, if you make antibodies against these viruses, those antibodies will cross-react with host tissues. But again, whether these play a role in bona fide autoimmune syndromes isn't known. Many viruses cause immunosuppression. That is, they suppress globally the immune response. They may replicate in some cells of the immune system. They could infect you at birth or during development, which would lead to tolerance. They can mess with the cytokine balance and signaling, or they can make proteins that interfere with the immune response. These are all mechanisms of immunosuppression. So here is an experiment where we are looking at the effect of measles infection on the tuberculin test. Remember, if you're infected with tuberculosis and you put a little tuberculosis antigen, you get a delayed type hypersensitivity response at the site of inoculation. So this is what we're measuring here, the millimeters of induration or swelling. So this is the baseline of an uninfected animal. And now this is a human being which is infected here and given the TB test. Now here is at the height of the rash of measles. This is how much tuberculin reaction you get. So you see it's very much suppressed compared to an uninfected person. And then as the rash goes away, you regain this delayed type hypersensitivity. So if you do a tuberculin test at different weeks after the rash is going away, you get a regular response. So that's immunosuppression. Somehow the infection has suppressed the cellular response that leads to this swelling caused by tuberculin. These are 3 examples of viruses that immunosuppress. Measles is a very, very serious immunosuppressing virus. It infects a variety of immune cells, and as you saw, it causes a reduced delayed type hypersensitivity, but more significantly, many people with measles get other infections because they can't fight them off. Rubella infects lymphoid cells and also leads to other infections, and of course HIV is probably the best known. It infects CD4 positive T cells, and you get susceptible to a variety of other opportunistic infections and cancer. So measles virus infects both dendritic cells and monocytes. So it compromises a very big arm of the immune response. T cells in people who have measles are down 50%. And you have aberrant cytokine responses. So these are some of the reasons why measles gives you so much immunosuppression. So the last bit that we'll cover today is susceptibility to disease, what genes control it, and what non-specific factors control it. So you want to identify genes that control disease, obviously, because if you, say, had a human gene that prevented infection with a virus, you could capitalize on that. So in mice, there's a gene called MX. If you take that away, they don't get flu. But as I said last time, mice normally don't get flu, so this is just a proof of principle. There is a gene in mice called FLV that makes them resistant to flaviviruses. And flavies include West Nile, dengue, yellow fever. So this is an interesting gene to study. This encodes an interferon-induced protein, 2', 5' oligo A synthetase, which you may remember works with RNA cell to degrade virus RNA. So if you have this gene, you can resist flavivirus infections. If you take it away, you're susceptible. Unfortunately, none of these play any role in human infections. And sometimes that's what happens. You get an exciting lead in an animal model, but it's not relevant in people. Here's an example of what this FLV gene does yes, sure.
We don't have the 2', 5' oligo A?
We have 2', 5' oligo A, but there is no association in humans of resistance to infection with that gene as there is in mice. So in mice, if you take away the gene, the FLV gene, you are totally susceptible to infection. And if you have the gene, you're resistant to infection. But in people, we don't have a similar correlation. Even though we have the gene, there's no example of it determining susceptibility. What do we have for people? Well, we don't have very much. It's very hard to find single human genes that influence susceptibility. If you think about it, this is an issue of population genetics, and it's very difficult to sort out. Now, pretty soon, we'll be able to sequence everybody's genome, because the cost now is about $1,500 per genome. All of your bases can be sequenced. And actually, you can get yours sequenced for free if you want right now. Personalgenome.org, I think. It's a project out of Harvard University, which is aiming to sequence 100,000 human genomes. So if we can sequence your genome and then see what infections you're susceptible to or not, we may be able to make some correlations. But right now, we don't have that. Most of the genes encode components of your immune system that control susceptibility. For example, the resistance to HIV in people lacking the CCR5 co-receptor. That's a very interesting resistance, but as I told you a long time ago, that mutation makes you more susceptible to West Nile virus. Certain people who are carriers of herpes have mutations in TLR3 or related protein that's involved in the signaling of TLR3, UNC93B. So this is, again, an immune response gene. And many susceptibility genes map to the antigen presentation genes, the major histocompatibility genes. So that makes sense because the ability to present a virus antigen to the immune system can be a major determinant. But these are immune system molecules. Is there anything else? Here's an example of MHC controlling virus susceptibility These are mice infected with LCMV. Two different MHC classes or haplotypes of the mice. You can see one is susceptible and one is quite resistant to infection. So the ability to present antigen is a major determinant of susceptibility. So we don't really have any non-immune genes that influence human susceptibility to viruses. We do have very many non-specific situations which may eventually be localized to specific genes, but they haven't yet. Age is a big determinant of infection. Typically, very young and very old people are the most susceptible. I'll show you an example of that, but it doesn't always go that way. Sometimes it's the opposite. And young people, their immune response hasn't matured yet, so they're more susceptible. But that's also a plus. They don't have as much immunopathology because they don't have as much of an immune response. So if you infect mice with LCMV in adult mice, it's lethal because they have a vigorous immune response and it's an immunopathological disease. And infant mice survive. Why are old people more susceptible to disease? Their lungs are less elastic. They have weaker respiratory muscles. The cough reflex is diminished. These are some of the determinants that probably influence susceptibility. So here are some examples of that. It's West Nile infection in rats. This is an experiment where they have used rats of different ages, which is on the x-axis from 0 to 18 days old. And this is the LD50, the amount of virus you need to kill 50% of the mice. So you see with very young rats, you need a lot of virus to kill half of them. But as you get old, you need less and less. So this is an example of where the older rats are more susceptible to infection. Age-specific replication of the virus in the brain. We're looking at 1.5 days, 16 days, and 3-month-old rats. So you can see here the young ones are more susceptible to virus replication in the brain. And as you get to middle age, 3 months is sort of like a middle-aged rat. The virus doesn't grow well. But if you did this with older rats, you might see an increase in replication. This is an experiment in humans, actually. This is an island population, the Faroe Islands, which is in the 1800s was relatively isolated, so it was rare that diseases were introduced frequently. And this is just a breakdown of the mortality caused by measles in 2 different time periods, a 10-year period here and then in a year later. And you can see very susceptible. The youngest are very susceptible, and also the oldest are quite susceptible, and those in between are less susceptible to infection. And again, we understand some of the determinants of this. Again, weak immune responses, poor respiratory responses, but we don't understand it all. In mice, as mice age, they become more resistant to neurological disease caused by Synbis virus. And we know why that is. That is because as mice age, they produce more of this protein BCL2, which has anti-apoptotic properties. So older mice make a lot of this protein, so they are more resistant to infection. Here's another example. Parvovirus infections of neonatal rats leads to replication of the virus in the cerebellum. But if you infect older mice, older rats with the virus, there's no effect. And that's because the virus replicates in cells of the cerebellum, which are dividing during the neonatal period. They stop dividing in the weanling animals, and they're no longer susceptible to infection. So that's another example of a mechanism. But as I said, there are age exceptions. Respiratory syncytial virus causes disease in infants, but not in adults, and not even in older people. And this may be a consequence in defenses that develop or susceptibility of the cells. In 1918, the pandemic was lethal for the very young and also in between, which is very unusual. Polio, mumps, measles are all milder at a younger age. So polio paralyzes older people more readily than younger people. We don't know why. Maybe there's a better balance of the immune responses at that age compared to other ages. So here, for example, is a curve of West Nile mortality in Israel in 2000. And you can see older people are more susceptible than the very young. And here's a graph of influenza mortality in 1918 compared to typical influenza mortality. So the very young and the very old are typically the most susceptible to influenza. But in 1918, in addition to those groups, you also had this peak of susceptibility in the middle. And we really don't understand why that is. And finally, other determinants. The gender has an effect on susceptibility. Pregnant women are more susceptible to infection. These may be hormonally related susceptibilities. Of course, malnutrition is a big determinant. Malnourished children are very susceptible to viral infections. And for that reason, measles is much, much more lethal in developing countries than it is in Europe and North America. Cigarette smoking really increases your susceptibility to respiratory infections. Stress, but remember, stressed is dessert spelled backwards. So if you're stressed, just have some dessert. I saw that in an ice cream store. Air pollution is another one that causes an increase in respiratory disease. And these are all things we really don't understand the mechanisms of. So maybe someday we shall. But so don't smoke, don't get stressed. And I'm sorry if this course stresses you, but and don't breathe bad air.